So Miguel, I have the pleasure to introduce you to Giorgio, but I think you already know each other.
Hello.
Hi. And then Gareth Owen, that is basically our cancer expert in Chile. If you talk cancer, if you say the word cancer, is Gareth Owen. So yes, he's here with us. And yeah, okay.
Great. So in the meantime, we are live streaming. So if you want to start to introduce our guest.
Okay. Okay, so hi everybody. Today I'm truly excited to have Giorgio Scita, a speaker at our DevMech webinars. Andrea and I had the pleasure to know Giorgio in the context of the Mechanobiology Institute in Singapore and at IFOM in Milan. And of course, at several conferences such as the Mechanobiology in Singapore and in Vietnam or Gordon conferences. Well, just to give you a brief idea about Giorgio, so Giorgio obtained his degree in biological science and the postgraduate specialization in food chemistry and technology at the University of Parma in Italy. Then for his postdoc, he firstly moved to Berkeley at the University of California and then to NIH. In the 1995, he joined the European Institute of Oncology in Milan. And since 2001, he has been a principal investigator at IFOM, that is the FIRC Institute of Molecular Oncology. And there he leads a group working on mechanisms of tumor cell migration. Well, he has authored more than 145 publications, which include more than 120 original articles and 12 invited reviews. And he serves as editor and reviewer for many impact factor journals. During his career, he has focused on combining molecular cell biology and biochemistry with other disciplines, such as signaling to actin dynamics, tumor cell biology and pathology, and in the last 5 years, biophysics of collective epithelial carcinoma to address fundamental questions on carcinoma progression. So today we'll share with us his current work on tissue fluidification in invasive breast cancer. So I thank you, Giorgio, for accepting our invitation, and the floor is yours.
Can I just something? Because I was reading your bio and I found something funny and something we have in common. We both had as a childhood dream to study ethology. And then we both and then both figure out that cells are equally interesting, if not more.
Yeah, that's actually going to be a theme of what I'm going to be telling you about the relationship with the animal behavior and as living complex set of entities.
Fantastic.
So that's cool. So thank you very much for the opportunity to discuss some of the data. I generally get bored with data that are published, so I decided to tell you something more related to what we are currently trying to publish and is not out yet on the theme that we've been focusing on the theme that we've been focusing the last few years, as Christina pointed out, which is the issue of phase transition at the tissue level, which is a phase transition between a solid and a fluid-like state in the context, in particularly of early invasive, actually early lesions that are becoming invasive breast cancer. And it has been, I don't need to tell you this, but the mechanical property of cell and tissue have become pivotal regulator of cell fate and in physiology and pathology. And during organ development, for instance, frequently cells and organs actually become crowded with cells to acquire solid-like state, which is thought to be essential for their development of physical, chemical, and functional property. On the other hand, let me see if it works. When a tissue needs, for instance, to pass through narrow gaps, to close a wound, or to disseminate, some level of fluidity needs to be gained. And so you can go from like a solid, more solid-like, and I'll get back to it as to what we mean about solid, to a more fluid-like tissues, and the fluidification actually facilitate all these processes. And one way to account for this phase transition is the notion that has been put forward, particularly thanks to the work of Jeff Fredberg of jamming transition, which is a phase transition from a solid-like to a tissue-like level, characterized by highly coordinated mode of locomotion, which is very similar. That was my ethological roots or interest to the type of motion that entities like sheep grazing into grass actually undergo. And this theme of mechanics or dynamics apply in the context of tumor evolution has become particularly prominent in the last 10, 15 years. This is a beautiful review that if you have missed it, I invite you to read by Jackie Goetz, where she pointed out that all the stages of tumor progression from the early tumor formation and invasion into the surrounding site to the extravasation, intravasation, and seeding of metastases requires tumor cells to be challenged and to face a number of mechanical properties. One striking example is offered by this work by Peter Friedel, where he has actually studied the early phases of dissemination when a tumor from a confined site becomes locally invasive by extending frequently, in the case of carcinoma of tumor derived from epithelial cells, collective strands and entity that move as such by coordinating their locomotion, as it can be seen here. And like condition like alteration of cell-cell adhesive property, which is a critical component of carcinoma of tumor that are derived from epithelial entity, and confinement exerted by the extracellular matrix impose changes in the mode of motility, which can be read through the length of a phase transition from a solid to a more liquid-like state. And we will argue that this phase transition may actually be one way to account for the acquisition of locomotion, of collective locomotory behavior, which is required for the early step of tumor dissemination and eventually tumor metastatization at distal site. And that again, to combine the work of physics with mechanics and dynamics with the ethology, a number of tools that actually are used right now to study collective cell motility are the ones that have been applied to study complex systems, like the one that gained to Giorgio Parisi the Nobel Prize in Physics, and he had spent some time discussing and understanding the dynamic of these flocking birds in how they move together, how they are coordinating their motion in order, in this case, to escape from predators. And we see that some of the tools that actually is developed are actually something we are applying to the study of collective locomotory behavior and phase transition from a solid to a liquid-like state. But the system that we are particularly interested in studying and apply the rules of phase transition is the evolution of a specific carcinoma, which is a human breast carcinoma. And here what you can see is a sort of evolutionary trajectory of this carcinoma, which invariably develop inside the luminal component of the mammary duct. And frequently, about, for instance, 20% of all carcinoma lesions are diagnosed as ductal carcinoma in situ. What this word means that this carcinoma typically grow within the mammary duct. They undergo, they can become particularly dense due to proliferation, which is a confined proliferation, and thereby they are experiencing a number of mechanical stress due to compression, excessive crowding, that actually impact their physical state, favoring potentially a transition from a liquid-like to a solid-like dense state. And in fact, about 70% of these early diagnosed lesions are actually benign in nature, in the sense that if left untreated, they will actually never progress to become invasive. Nevertheless, about 25 to 30% of those will actually be able to overcome the caging and the tumor suppressing environment exerted by this excessive compressive crowded state, acquire liquid-like property, as we would argue, and acquire local invasive property through the extension of collective locomotory strands, cords that invade into the surrounding matrix. And so to try to understand tumor evolution with the length of biophysics, here we need to understand what are the rules that drives the solid to liquid-like transition, whether solid to liquid-like transition, as I would argue, is a sort of like alternative gate to cell migration that enables this early lesion to become locally invasive and eventually to seed metastatic disease. And one way, as I turned out, one way to try to make sense of all the biochemical and biophysical interaction that cells establish among themselves and with the extracellular environment is the notion of cell jamming or cell unjamming, as I would call. And the best way that I had to describe the phenomena that I'm going to be discussing is depicted in this movie. What you see here, and I'll run it in a second, is a model monolayer formed by normal mammary epithelial cells that are seeded very densely to cover the entire space, almost like tiles occupying the space of a floor. And then as the density rises, I mean, at the beginning, when the density is not so dramatic, cell-cell interaction motility is allowed. The system actually behaves like a liquid where individual cell component actually can exchange. But as the density increases due to proliferation at a critical precise density, the tissue stops to be fluid, becomes each individual cell become caged by its neighbor, and the tissue acquire what looks like solid-like property. And indeed, we can actually provide quantitative measure of this transition from a liquid, in this case, to a solid-like state by, for instance, applying particle image velocimetric analysis, which is what is seen and quantified here, which enables to measure velocity vector across the entire field of view. It also allows you to measure velocity in local region within the monolayer. And this is exactly what the coloring code is indicating, whereby in yellow color or warmer color, it indicates region of the monolayer that move with the same direction and the same velocity vector, if you wish, of the entire field of view, while in blue you have like region that move in the opposite direction, which capture one of the critical feature of this system that are out of equilibrium, but transiting from, in this case, liquid to a solid, which is the emergence of dynamic heterogeneity. All right. And so one of the questions, just like to summarize what we are dealing here with, I make use of a movie that is taken from Jeff Fredberg, whereby a solid-like would need is a system where like there is an energy barrier that has to be paid in order for neighbor to exchange their position. And this is primarily due to the fact that there is a local tug of war between cell adhesive property and tensional junction. And when, for instance, increased adhesion, reduction of tension enables tissue to acquire liquid-like, where liquid here is intended once again with the ability of each individual cell to move with respect to its neighbor. And if you look at the dynamic of each individual cell within this tissue, you can clearly observe a transition from an immobile to a sort of reawakened, collectively moving, as I will argue, fluid-like system. All right. Why this type of notion is particularly interesting and catching in the context of tumor evolution, but in general in development? The reason being is that we can describe collective entity in our discussion, epithelial and tumoral epithelial entity being regulated in terms of its dynamic transition state by a set of simple parameters that govern the transition between a solid and a liquid. Parameters like the strength of cell adhesion, less obvious parameters like cell shape and shape variance, and individual cell locomotion. And that actually will design a sort of like three-dimensional phase diagram where each individual collective entity can switch from a solid to a liquid based on changes in simple biochemical and biophysical parameters. Is this relevant for the evolution toward normal epithelial cell? It appears to be essential to transit versus a solid-like state that enables to confer to the epithelial tissue also the rigidity property that each epithelial organs and glandular organ in particular would have, but it may also be an extraordinary way to act as a tumor suppressive environment, because in condition of overcrowding of solid-like state, the emergence of each individual clone that would need to move around is certainly suppressed. And indeed, there are beautiful work done a few years ago now, these two Nature paper where actually they studied ERBB2 model system. ERBB2 is an oncogene that is a major driver of aggressive breast carcinoma. And when they look at late ERBB2 lesion, they realize that these are packed, dense, and actually very poorly capable of seeding distal metastasis as compared to what? As compared to early lesion where local cell rearrangement and fluidity-like property actually enable efficient metastatic seeding. And then there is an additional feature of this system that are transiting, which is depicted by this set of movie where you actually the only thing I pointed out here is like a solid-like system is characterized by reduced motility, reduced fluctuation in density and in volume, while the counterpart of a fluid-like system, there are large fluctuation in cell density and cell volume. And I'll come back to the importance of this fluctuation as the system goes from a solid to a fluid-like state. All right, so one catchy proposal is that we can utilize the jamming to unjamming or solid to fluid-like transition as a sort of, as I pointed out, gateway for tumoral cells that to gain what? To gain ability to move collectively into the surrounding matrix. And that is particularly interesting because you need to change few features of the system to switch the state from a solid to a fluid invasive, potentially invasive system. So the question we have been trying to address in the past few years is what are the molecular determinants that enables tumor cell entity and in general epithelial entity to regulate multicellular dynamic and this jamming transition. And I have to say that we stumbled upon one of the regulators totally serendipitously. We have been studying for a while a mechanism that regulates actin dynamics and how to control polarized actin dynamic in the context of single cell migration. And one of the critical molecules that we've been focusing on is ROB5, actually specifically ROB5A, which is a master regulator of endosomal biology. And here, for the physicists, the only thing to learn and the only name that I provide of a protein is, again, ROB5, which is essential for the generation of this early endosome, which can be viewed as a sorting station where any internalization pathway will end up. And here a decision needs to be made as to whether the cargo that is internalized recycled back to the plasma membrane or to different location of the plasma membrane, or it gets degraded via lysosomal degradation. And the internalization recycling cycle, we have shown in the past that are sufficient, for instance, to polarize actin polymerization signal for propelling membrane protrusion, enabling local directed locomotion, or to continuously increase or provide metalloproteases which can degrade the extracellular matrix, making a tumoral cells particularly prone to become locally individually invasive. But the surprise came when we started to observe the effect of the expression of this endocytic gene, which is upregulated in breast carcinoma and correlate with poor disease prognosis in the context of a normal mammary epithelium. As I pointed out, we seeded these cells at a very high density. There's no space. The entire field of view is occupied by cell. And then the system, the control are jammed. Then we genetically modify the cell to express in a doxycycline-inducible fashion RAP5. And the surprise came when we turn on RAP5, immediately the system starts to reawake its ability to move. They start to move in a very persistent, directed manner. And in fact, you can measure via either the root mean square velocity, which is a transient increase in velocity upon you turn on RAP5 expression, to recapitulate what we might see in human cancer, human breast cancer. And one of the things that was immediately clear, that the type of locomotion this collective entity would undergo is a coordinated collective locomotion, where the entire field of view, or the entire field of cells within the field of view, moves in a coordinated manner. And that can be captured and quantified by the correlation function of the velocity vector as a function of the distance between the two objects. And then you can calculate the length of correlation, which is a parameter that tells you how large is the pack of cells that move in a coordinated fashion. So we are witnessing a situation where the simple expression of an endocytic gene is sufficient to reawake the locomotion of an otherwise jammed solid monolayer and lead to the acquisition of a flocking, I use this word not by chance, it's a flocking fluid locomotory system. And that one gene is sufficient to elicit all this. And so the question became, how the hell does this can happen? And here I'll summarize a lot of work that we have been done in the last few years. If you have questions in the end, we can discuss about the detail of this work. But basically, a system that in which we can actually measure individual cell locomotion by labeling this monolayer with H2B GFP, and that, for instance, allow to capture this ballistic mode of locomotion when we measure the mean square displacement of each individual nuclei. So it's characterized by this large coordinated flocking fluid mode of motion, which is the results of the effect that RAP5 exert on a variety of different processes. So it's a critical regulator of the endocytic machinery, and as such, it affects junctional turnover. And so since we have long-range locomotory behavior, coordinated locomotion that spans 1 millimeter in size, one critical factor is the strength for cell-cell adhesion. And indeed, RAP5 affects junctional turnover. It affects junctional shape and morphology, increasing cell-cell surface contact, while enabling local cell rearrangement, because the increased turnover enables neighbor exchange. In addition, we constantly observe large density, which we call giant density fluctuation, and volume fluctuation in this system that are requiring locomotory behavior, and each individual cell is capable of extending protrusive activity underneath the neighboring cell and to reorient their locomotion with respect to their neighbors. And in fact, we actually can capture the complexity of the system by a simple vertex-like model, which has two key ingredients. One is a shape factor. I told you that shape actually can predict whether a system is solid or liquid, or whether there is neighbor exchange. And so shape, which is captured here by the perimeter versus square root of the area, is one of the critical ingredients that we have introduced in the model. And there is a sort of a magic number, this target shape, which is 3.81, predicted by theoretical work by Lisa Manning, Max B, and Jeff Fredberg, whereby a system that is below this 3.81, which is close to an hexagon and pentagon, is solid. Above, neighbor exchange is allowed, and thereby it's liquid. But we can capture what happens dynamically in the RAP5 reawakened locomotory behavior by introducing a zigzag-like realignment, the ability of each individual cell to realign its locomotion and the time that it takes. And under those conditions, we can identify a region which we call flowing liquid, which is depicted here in this movie, where actually cells actually exert large attraction. They move in a coordinated fashion while keeping on exchanging their neighbors. All right, so then we ask, is this system driven by a specific molecular set of mechanisms? And one that we have identified is depicted in, again, and summarized in this slide. What we one of the things that happens upon, one of the things, because many things that might happen upon elevation of an endocytic master regulator like RAP5 is an increase in number of endosome size, and an increase of a non-cluttering, unconventional way of internalization. And EGF receptor, whose activation is essential for this type of locomotory ability, becomes largely internalized into this increased number of endosomes. And this increased number of endosomes, which are packed with EGF in a sort of like quanta way, enhance localized signaling. And the signaling that is emanating from this endosome is a signaling that eventually talks to molecules that are essential for the actin polymerization machinery to generate protrusive activity and this cryptic lamellipodia that reorganize their locomotion and enables to reawaken what is gem and solid into a collective flocking locomotory mode. So then, since we are a cancer biology institute, I'm particularly interested in the context of the progression of ductal carcinoma in situ to invasive carcinoma. We start playing around with the ductal carcinoma in situ model cell lines that once grown in low attachment, they form beautiful spheroids or organoid-like. They're not really organoid because they're from cell lines, which we label with LIFACT and M-cherry H2B to be able to monitor the dynamic of the spheroid. And again, we can use the trick of activating RAP5 in this spheroid that are embedded in a thick collagen matrix, have one indication of this. And we notice that the RAP5, which is sufficient to induce this flocking locomotory mode, leads to persistent rotational motion, alteration in fluidity, which is particularly prominent at the periphery, which eventually exert, and I'll show you an example, large traction force on the extracellular thick collagen matrix in which these tumors are embedded to enable the generation of space where collective locomotory behavior takes place. We can actually provide evidence that occurs in ex vivo tumor slices, which are kept at air-liquid interface to be able to monitor their locomotion. And again, if we turn on RAP5, we start seeing this flocking collective locomotory mode of motion from a system that is otherwise solid and jammed. And that in vivo, in a mouse model, we can actually monitor the transition from a ductal carcinoma in situ, which is marked by this myoepithelial layer that confine the tumors, and then eventually this myoepithelial layer is lost to as the tumor becomes overtly invasive. And in the case in which we express a dominant negative RAP5 to impair RAP5 activity, we severely delay the transition from a ductal carcinoma in situ to an invasive fluid-like, we would argue, carcinoma. And that can be observed in human tissues. We have a battery of ductal carcinoma in situ with local infiltrative area that we have done together with the Claudio Tripoddo, the pathologist with whom we collaborate extensively. And what you see here is the expression level of RAP5, which is heterogeneous in nature. It becomes prominently elevated right at the site where collective invasive buds, potentially fluid-like structures, invade into the surrounding matrix, leading us to propose the fact that flocking fluid locomotory behavior enabled to overcome the caging imposed by the crowded cellular landscape of the ductal carcinoma in vivo to allow them to invade locally. And as I told you, these spheroids do exert large traction forces. And so here we actually monitor in a collagen thick matrix, which we introduce fiducial fluorescent marker, the extent of protrusion that this persistently rotated organoid upon induction of RAP5 exert on the extracellular matrix, and this traction is sufficient to remodel the organization of the extracellular matrix in which these spheroids are embedded. This is a second harmonic generation signal to monitor the type of collagen organization which we introduce ectopically, obviously. And it is obvious that upon RAP5 expression, not only they start rotating, here you don't see, but they dramatically remodeled extracellular matrix. They generate gap and channel into which collective locomotory strands and Indian file are advancing. All right. So then comes the part that that is not published yet. And we ask a simple question here. So now all this is rather satisfactory from a biophysical point of view. We are in the presence of something that goes from a solid like state, potentially to more suppressive like state, to a fluid, highly plastic collective entity that enables these tumors to acquire local invasive ability. We ask whether the associated reawakening of locomotion that is underlying this transition may actually impact on long-term consequences. When I say long-term consequences, I refer to the fact that there might be a set of veritable changes, transcriptionally driven, that change the state of a collective entity that is switching from a solid to a liquid-like state. And this, it's apparently a complex question, but then the type of experiment that we immediately did, that for a while caused us some thought as to how to interpret, was to compare the transcriptional differential profile of gene expression in control monolayer that are solid in nature versus RAP5 expressing either normal mammary epithelial cells or oncogenic models that recapitulate the ductal carcinoma in situ. And the differential gene expression was very striking. Immediately, gene ontology terms revealed that the most deregulated pathway were of the interferon or the innate immunity type of pathway, to an extent that was very surprising to us, because if we look at some of the core genes that define this interferon, or as I argue in a second, cytosolic DNA sensing pathway, are upregulated following reawakening of cell locomotion in normal or oncogenic cells and in a variety of different cell types by 1,000-folds. So there is a dramatic rewiring of the transcriptional profile, which coincide with the transcriptional profile, which has been associated to the ability of cell to sense cytosolic DNA. Is the so-called cytosolic DNA sensing pathway, which shares a number of these core genes, which are also the genes that you will find activated upon stimulation of the interferon alpha-beta pathway or activation of innate immunity pathway. All right, so that caused us a lot of nightmare, because I could not understand how to interpret this to occur in a purely epithelial system. These are pathways that are typically activated in response to viral infection, excess DNA, like in pathology, like the lupus erythematosus, that activate innate immunity like type of response. The fact that it was a cytosolic DNA sensing pathway was the first clue, because one of the best sensor of cytosolic DNA that has been characterized in the last 6 to 8 years is the so-called cGAS-STING pathway. This is based on the fact that cGAS, which is an enzyme that converts ATP-GTP into a D-nucleotide, which is called cGAMP, that becomes a second messenger. It does so by binding specifically to double-strand DNA that is present, accumulating in the cytosol. And that evolutionary, it's a system that has been evolved to fight viral infection, for instance, where the excessive amount of DNA is eventually released into the cytoplasm, activate the pathway that eventually leads to an interferon-like response, that is evolutionary evolved to fight viral infection. For what matters here is that the cGAS as a sensor is also it has a very high affinity for double-strand DNA, particularly if it is damaged, but we can discuss this later. And once it's activated, activate this STING, which stands for stimulator of interferon pathway, which is generally localized in the endoplasmic reticulum, but upon binding with the cGAMP, relocalize it to the Golgi, where it activates a set of kinases like TBK1, IKKB, which eventually phosphorylate the critical transcriptional factor that act as an interferon-dependent transcriptional regulator, which is IRF3, that upon being phosphorylated translocate to the nucleus and activate all the core set of this gene that we found they're regulated in these flocking locomotory fluid-like cells. And one thing that is important, so we notice that if we compare single cells versus dense monolayer, whether they're dense monolayer or they are actually, we observe the same thing in the context of spheroid or even in the context of tumor implanted into the mice, we have the dramatic upregulation of an interferon-like cytosolic DNA response, which becomes dramatically elevated once the cells are in contact with each other, suggesting that this is, again, an emergent property of a collective entity. We do see some upregulation in single cells, but the level and the extent of upregulation becomes 100-fold higher once the system is collective, and under those conditions, the system will be flocking fluid locomotory system. So then, is this pathway the one that is at stage and is the culprit of the dramatic transcriptional rewiring that we see under this condition? So we address this by the obvious, by hitting various key steps of the pathway. These are frequently enzymes, so there are available very potent and specific inhibitors, pharmacological inhibitor, like the RU521 or the H151 that target the activity of STING. But we can also, for instance, by molecular genetics, impair the expression of either cGAS, STING, or downstream pathway like IRF3, or even more downstream pathway like IRF9 and STAT1 and STAT2, which are all markers of inactivated cytosolic cGAS-dependent, cGAS-STING-dependent pathway. And here is the type of results that we typically get. RAP5 activate all these key set of genes, which are dramatically inhibited in their expression whenever we interfere with this pathway component, indicating that indeed the flocking fluid locomotory behavior induced by endocytic perturbation activates cGAS-STING dependent cytosolic DNA response pathway, which raised the question how this might happen, because that is really by mechanistically what we have been trying to address. And then the clue to how this might happen came from this older experiment that we have done when we look at the behavior of each individual cells once a system becomes flocking fluid locomotory in its nature, once it transitions from a solid to a fluid-like state via flocking fluid locomotory behavior. And what you see here is a normal monoepithelial cell that are engineered to express E-cadherin as a GFP marker, so a junctional molecule, which enables to track the position, motility, size, area of each individual cells, which is exactly what has been done here by Wei Miao Yu in collaboration with the Wei Miao at the Star Institute in Singapore. And it was very obvious, if we remove, as it is in this case, the global locomotion, that the cell undergo large fluctuation. They undergo fluctuation in size and volume that are, we expected to actually exert some kind of mechanical stress that is transferred from the global epithelial entity to each individual cells. And actually, the stress is transferred directly to nuclei. So here, there is a pipeline that Fabio Giavazzi and Stefano Villa, they are two biophysicists we basically operate as an extended lab, set out to measure in large number of nuclei, and thereby providing very robust data, the type of stress and deformation that once a system becomes flocking fluid exert on the nuclei. And I just zoom out on some of these because it becomes very obvious what happens. The extent of deformation, of change of shape, RAP5 flocking fluid locomotory cells undergo is dramatically increased. Their speed is enhanced. And in fact, we can calculate by measuring the rate of mean square strain change, the strain rate, which is significantly elevated in the case of RAP5, suggesting that the flocking fluid locomotory system eventually exert mechanical stress that are transferred to the nuclei. Why I'm stressing this point? I'm stressing this point, and I give you an additional pieces of evidence, because we notice by looking at the RNA-seq that one critical determinant that is changed in the nuclei of this flocking fluid locomotive behavior is lamin B. Lamin B, well, lamin AC are the major nuclear envelope intermediate filament protein that confers rigidity and resistance to nuclei to mechanical type of stress. And so we observe the combination of this mechanically altered shape-deforming nuclei in the presence of a weakened, potentially weakened nuclear envelope. So condition that actually may suggest that the nuclear envelope is prone to rupture, to release cytosolic DNA that may actually be sensed by SIGA sting and activate the response, the cytosolic DNA response pathway and interferon-like gene pathway. And so we set up a bunch of experiments. I show a few to see whether that was the case. One exploited the ability of SIGAs to bind specifically cytosolic DNA. And so we proved that that is the case by illuminating and breaking through localized laser illumination the nuclear envelope. And here what you can see that the SIGAs expressing cell that typically is localized cytoplasmically in interface cells becomes prominent and rich right at the site where we induce a nuclear envelope break. And so we utilize the accumulation of this perinuclear SIGAs foci as an indication of the fact that the nuclei in flocking fluid locomotory cells are prone to undergo rupture and activate a SIGAs sting pathway. Indeed, the number of pericellular nuclei of perinuclear foci becomes significantly increased, specifically under condition of flocking fluid locomotory behavior, not when individual cells are monitored, and the activity of SIGAs is elevated, and we can test this by measuring in these monolayers the level of the product of SIGAs enzymatic activity, which is SIGAMP. We further did a bunch of other experiments. For instance, we tested, and I'm not going to show you this, whether the cells that actually undergo this type of rupture, once if we monitor a NLS localized GFP, actually leaks out GFP, and that's the case. And additionally, we establish a protocol to directly observe potential nuclear envelope rupture by using CLEM tomography, correlative light electron microscopy tomography reconstruction. To this end, we express GFP SIGAs, focus on those regions perinuclear accumulation of SIGAs, and analyze the same region by correlative light electron microscopy to observe a couple of things. With the black dots, you see the detection of SIGAs, which is accumulating right where the chromatin is condensed. And at the site of this chromatin condensation, there is a communication between the cytoplasm and the nuclei, which enables SIGAs to recognize chromatinized cytosolic DNA and being activated locally at the site of nuclear envelope rupture. We can do that by immunofluorescent type of analysis. In some cases, we see dramatic perturbation of the nuclear envelope and the localized activation of SIGAs right at the vertex, where the cytosolic DNA might actually leak out, leading to the activation of this enzymatic activity. There is one additional set of things that is related to mechanobiology. So because we start reason that what we are looking at when we induce this flocking fluid locomotory state is more maybe a situation where the epithelial entities undergo a sort of chronically mechanical stress state, which is very similar to a chronically stretched state, where if that's the case, you would see changes in expression of lamin protein, which we did see, but also alteration in the nuclear, in the perinuclear actin, which frequently have been associated as an attempt of cells that are undergoing cyclic stretch, for instance, to protect the integrity of the nuclear envelope by generating a cage around the nucleus itself, alteration inside the nucleus of chromatin state, which is associated with alteration of nuclear rigidity and increased nuclear size. So we actually, all these were actually very much inspired by this beautiful work by Sarah Wickstrom that subjected to persistent chronic stretch a set of epidermal stem cells and study the behavior of this. And she noticed enlarged nuclear size, increased nuclear rigidity, increased alteration in heterochromatin state, formation of perinuclear actin ring, all events that somehow are interpreted as an attempt of cell to protect the integrity of the nuclei, but eventually, upon persistent existence of this mechanical stress, things fails, DNA gets damaged, and then you have release of cytosolic DNA. So we tested whether we were in the presence of all these potential alteration. And indeed, the nuclear area is significantly increased in these flocking fluid locomotory cells. There is the persistent and dymatic remodeling of the actin cytoskeleton with the formation of this prototypical actin ring that is persistent in this flocking fluid locomotory behavior, flocking fluid behaving cells, and the chromatin is significantly altered with an increase of H3 of constitutive H3K27 trimethyl chromatin, which is frequently associated to an attempt to increase rigidity and resistance of nuclei to persistent mechanical stress. In addition, I can skip this, we set out to test whether indeed all this would actually lead to an increased rigidification of the nuclei. And the first set of experiments were done by atomic force microscopy. We know that it's very difficult to assess whether nuclei within a monolayer are more rigid, because it's very hard to access directly the mechanical property of a nuclei. So one way, indirect way, was to measure by atomic force microscopy the rigidity of the cell right in the region above the nuclei, which is significantly increased. That was okay, but we know that there is a contribution of the actin cytoskeleton of the nuclear of the membrane tension. So we set out to measure a bunch of other things. For instance, we subjected cells to a pre-stretch and then measure by looking at the variation on the area, the extent of strain these cells undergo, and if, and actually focusing on the nuclei, and if a nuclei of these flocking fluid locomotory cells is more rigid, you would expect to undergo less area change strains in the end, which is what we see. Then we subjected cells to pass through a predefined confined channels, and we study the type and the extent of deformation and the time it would take a cell to deform their nuclei. And so we actually plotted then the mean aspect ratio, which is a quantity that is, well, there is no really, the aspect ratio was actually the best one to test, and we could observe the rate of aspect ratio changes, which is significantly delay in the case of R5, suggesting once again that the nuclei are more rigid. We also did one additional couple of experiments where we actually measure the what is defined as the excess of perimeter. So if we can measure the perimeter of a nuclei and we assume that there is a perfect circles in this case, we can then determine what is the difference of the excess of perimeter if a system or a nuclei, a floppy nuclei, it's characterized by a lot of convoluted surfaces, then it would be closer to 0. If it is very rigid and close to, it would be close to 1. Yeah, so 1 would be an excessively folded, possibly flocky nuclei. 0 would be a completely rigid system. And indeed, when we look at the nuclei of Rab5 versus control cells. They are larger, but also their nuclear envelope. This is done by staining with SAN2, and in our nuclear envelope protein is significantly close to 0, indicative of a more rigid system. Finally, and then I'll stop this, we look to see whether the fluctuation of the nuclear envelope were affected by the expression of Rab5. We did it by this linear plot where we can actually measure the intensity profile of a Nesprin GFP, which localized to the nuclear envelope, and study its fluctuation around the peak. And then again, we actually run movies of this kind. And again, we assume that a rigid nuclei might actually undergo less fluctuation with respect to a more floppy one, which is exactly what we observe. All right. We also measure by beryllium microscopy, and I didn't put it here, and we obtain similar evidence that actually the nuclei of Rab5 expressing cell rigidify to try to cope with the increased mechanical stress. Nevertheless, they fail to protect the nuclei. And then when we measure DNA damage, we see a significant increase in gamma H2X, which mark foci of DNA damage of P53BP1. We also measure by comet tail assay the extent of damage that these nuclei undergo. All right. So just to summarize, and then I can stop here. So what we are proposing is that following the reawakening toward flocking fluid motile monolayer, there are a bunch of events that cause mechanical stress that are transferred to the nuclei, which undergo large deformation. The nuclei tend to resist this by mounting a mechanoprotective event or set of response, which include actin information, chromosome condensation. They increase the nuclear area. That eventually fails, leading to nuclear envelope rupture, frequent nuclear envelope rupture that fails to be repaired, leading to the extrusion of cGAS, of cytosolic DNA, which is sensed by cGAS and activated transcriptional response. All right, and an IRF3. Is this relevant? Is something that we can see in a pathological system? And we did a couple of experiments using either mouse xenograph, where we can introduce ductal carcinoma in situ, generate a ductal carcinoma in situ, and see whether some of the markers that we have identified following this mechanical perturbation are elevated, and analysis of human ductal carcinoma in situ. And so, for instance, if we measure cGAS, we observe only in the Rab5 xenograph the prototypical accumulation in crescent-like or perinuclear foci of cGAS, which are otherwise is not activated. We see also elevation of gamma H2X only in this xenograph expressing Rab5, and the perinuclear accumulation of gamma H2X is very similar to what we observed in ductal carcinoma in situ from human patients, which display local invasive regions where the perinuclear accumulation of cGAS, either via formation of dot or crescent, is elevated. And then with our pathologists, we actually measure and analyze the expression of Rab5 in a variety of ductal carcinoma in situ with local invasive region, and invariably we observe a deregulated gradient-like expression of Rab5 right at the site where eventually there will be an invasive foci, which is accompanied by cGAS elevation and gamma H2X activation. And I can skip this. We have done experiment in organoid, but I can come back. Let me summarize what I told you about. So we are basically connecting mechanics, DNA damage, and cGAS signaling. Mechanical perturbed solid-like can undergo fluidification, and that occurs through dramatic elevation of motility, which exert mechanical stress that is transferred to the nuclei, which becomes eventually, despite all the mechanoprotective event, prone to be rupture with elevation of a cGAS interferon-like stimulating gene transcriptional program. What we are currently doing is to understand whether this transcriptional change of fate of this tumor is either proto-murigenic or pro-immunogenic. We have some evidence that this system or this situation actually facilitate the emergence of traits like chemoresistance and a mesenchymal-like transition, which would suggest that are proto-murigenic. But we also entertaining the possibility that by reawakening a solid-like tumor into a fluid-like one, the activation of the innate immunity CGAS pathway may actually render this tumor from being immunologically cold to immunologically hot. And this is something we don't have any evidence yet, but we are currently investigating. So just to summarize, and then I finish, I told you today a story that was totally serendipitously where an endocytic gene is sufficient to unjam what is otherwise solid, static, rigid, and jam. And that enabled the escape from a tumor suppressive environment, the acquisition of flocking fluid locomotory behavior, which is instrumental for its invasive ability locally, to the change of transcriptional state by activation of CGAS pathway that may endow these cells with metastatic traits that are an advantage once the cells need to seed distal metastatic site. And this work, as you might imagine, is a concerted effort from many different groups. So the people in the lab that actually contribute mainly to the work is the work by Chiara that now is gone, and in particular by Andrea and Emanuela, but everyone in the lab contributed. We have a long-standing, we really operate as an extended lab with a group of biophysicists or physicists led by Roberto Cerbino until a few months ago, and now by Fabio Giavazzi and Stefano Villa contributed critically. We have a long-standing collaboration with the electron microscopy in IFOM, with the pathologists in University of Palermo, Claudio Tripodo. Bioinformatic unit of IFOM has been instrumental to identify some of the events, and I'll thank all you for your attention and the agency that supported the work. Thank you very much.
Great, thanks a lot. Very fascinating work. But there are a lot of questions. I know Gareth needs to leave for another meeting soon. So yeah, go ahead.
All right, thank you. As I'm first to speak, thank you. That was a wonderful talk. I learned a lot. Unfortunately, I'm not a mechanobiologist, so my question is going to be extremely basic. But I was just wondering if you could explain to me a bit better this concept of flocking, because what I'm wondering is flocking and metastasis the same thing, or two separate entities, or two related entities? I got the impression that all the cells, when there's not a compact tumor, all the cells are moving all the time. So my question is this something characteristic of cancer, or is this something characteristic of normal tissue that is not undergoing development? So this whole idea of the moving. So the question is, these processes you're talking about, do they occur when the cells start to get isolated and they actually can migrate away, or are these occurring within a tumor where E-cadherin is connected, and it's just part of the general physiology of a tissue that there's this constant flocking, constant moving?
All right, so this is a very good question. I can try to summarize some of the question, because this is something that has always we always ask ourselves. So is this one way to frame this from a very basic point of view, is whether this transition is related to density, for instance, to the number of cells that are present in an entity. And what we have seen is that this transition is totally density well, it's not totally is largely density independent. It requires strong cell-cell adhesion to occur. So within a normal tissue would actually tend to become solid, where flocking fluid locomotory behavior are very, very much limited, unless you introduce a wound, for instance. Wounding would enable the reacquisition of flocking fluid motility, unless they need to reproliferate, and then the acquisition of fluidity appears to be a prerequisite for migration to occur. So let me also add one thing. So there are work done by Daniela Vinjevic that actually using time-lapse microscopy monitor the motility of each individual cell within a tumor. And she observed like this constant relative limited motility within the core of the tumor, which is associated with their ability to invade into the surrounding matrix. Now, you also asked the question, what is the relationship between tissue fluidity, flocking fluid locomotor behavior, and metastasis? Well, I mean, there's a long road from local invasive tumors to metastasis. But we can argue, or you can argue, or we can argue that the early transition from a solid entity like a ductal carcinoma in situ to an invasive, locally invasive, is the first step toward the metastatic dissemination. For instance, RAB5 is particularly prominent in lymph node metastasis compared to primary match tumors or in distal metastasis, and correlates with distal metastatic dissemination. Whether it is causally related to that, it's more difficult to address.
Okay, thank you very much. Thank you.
Great. Christina?
So yeah, great talk. I just wanted to ask you, because you just mentioned now that this process is density independent, but I think I remember having read some publication about RAB5 being also involved in proliferation, right? I mean, connected to the cell cycle and so on. So I'm just wondering, I mean, if you have checked also the effect on RAB5 on proliferation.
All right.
Yeah. So, yeah.
All right. So the work you're mentioning is related to the fact that RAB5 appear to be required for proper mitotic cell division, and that those experiments were all done upon perturbing the expression of RAB5 A, B, and C. So what we see an effect on proliferation by the ectopic expression of RAB5, which is maybe it also can be observed if you look at the transcriptional profile, because all the mitotic genes are downregulated. Indeed, we see a decrease in cell proliferation. So what I'm saying is that in this dense monolayer, number one,